Today, an FDA breakthrough therapy designation in lung cancer, encouraging results with CAR T-cell therapy in non-Hodgkin lymphoma, disappointing results in myeloma, a promising combination in follicular lymphoma, and highlights from the 2016 NCCN Hematologic Malignancies Congress. Welcome to Enclave News Network. I'm Gina Columbus. The FDA has granted a breakthrough therapy designation to alectinib as a frontline treatment for patients with ALK-positive non-small cell lung cancer, according to Genentech, the manufacturer of the ALK inhibitor. The designation, which is meant to expedite the development of promising new therapies, was based on findings from the Phase 3 J-ALEX study, in which alectinib reduced the risk of disease progression or death by 66% compared with the current first-line standard crizotinib in patients with advanced or recurrent ALK-positive NSCLC who had not previously received an ALK inhibitor. The FDA granted an accelerated approval to alectinib in December 2015 as a treatment for patients with metastatic ALK-positive NSCLC following progression on crizotinib. The ongoing Phase 3 ALEX trial is comparing alectinib with crizotinib in the first-line setting for patients with advanced ALK-positive NSCLC. Results from the ALEX trial are intended to support converting alectinib's accelerated approval into a full approval that will also expand the indication to the frontline setting. In non-Hodgkin lymphoma, treatment with KTE-C19 demonstrated a 79% objective response rate and a 52% complete remission rate in patients with aggressive chemorefractory disease. In the Phase 2 ZUMA-1 study, the therapy was explored in 51 patients with diffuse large B-cell lymphoma and 11 with either transformed follicular lymphoma or primary mediastinal B-cell lymphoma. In the DLBCL cohort, the ORR was 76% and the CR rate was 47%. In the TFL-PMBCL group, the ORR was 91% and the CR rate was 73%. Overall, 39% of patients with DLBCL who received KTE-C19 had responses for at least 3 months, while 33% had a CR for that time frame. In the TFL-PMBCL cohort, the 3-month ORR and CR rates were both 64%. Grade 3 or higher cytokine release syndrome and neurological toxicity were observed in 18% and 34% of patients, respectively. 2 patients died from treatment-related adverse events. The anti-CD19 chimeric antigen receptor positive T-cell therapy has received breakthrough therapy designations as a potential treatment for DLBCL, TFL, and PMBCL. According to results from the Phase 3 CLARION study, carfilzomib did not show a progression-free survival benefit over bortezomib when combined with melphalan and prednisone as treatment for transplant-ineligible patients with newly diagnosed multiple myeloma. The results show that the median progression-free survival in the carfilzomib arm was 22.3 months compared with 22.1 months in the bortezomib arm. In an early assessment, it was found that carfilzomib also did not show superiority over bortezomib for overall survival. Moreover, the rate of fatal treatment-emergent adverse events was higher with carfilzomib at 6.5% versus 4.3% respectively. Grade 3-4 adverse events were observed by 74.7% of patients in the carfilzomib arm versus 76.2% who received bortezomib. Amgen, the manufacturer of carfilzomib, stated that it plans to submit full data from CLARION for future presentation and publication. In patients with early-stage, low-grade follicular lymphoma, treatment with chemoradiation demonstrated a significant improvement in progression-free survival when compared with involved field radiotherapy alone. A multi-center randomized trial showed that patients who received chemotherapy plus IFRT had a 10-year PFS rate of 59% versus 41% in those treated with radiotherapy alone. Additionally, a trial protocol amendment allowing rituximab to be added to chemotherapy did have an impact on the PFS benefit. The trial, which was presented at the 2016 ASTRO annual meeting, also showed that overall survival was prolonged in both arms. Patients who were randomized to systemic therapy had a numerically superior OS at 10 years, at 95% versus 86%, but this did not achieve statistical significance. However, systemic therapy also had a major impact on the occurrence of high-grade histologic transformation, as 10 patients treated with IFRT alone had disease transformation to high-grade lymphoma versus 4 who were given the combination therapy. The 2016 NCCN Hematologic Malignancies Annual Congress was held in New York City over the weekend, highlighting some new agents on the horizon, as well as the incorporation of the latest approved therapies into treatment guidelines. Specifically, in multiple myeloma, experts discussed the primary therapy for patients to be induction therapy with a triplet regimen of bortezomib, lenalidomide, and dexamethasone, followed by autologous stem cell transplantation, consolidation, and lenalidomide. For the second-line setting and beyond, the NCCN incorporated panobinostat, ixazomib, daratumumab, and elotuzumab into the guidelines. In acute myeloid leukemia, researchers explained that the landscape is expected to change significantly. While the standard of care approach has been 7+3, novel agents are poised to transform the fields, including venetoclax, CPX351, and midostaurin. All three of these agents have been granted breakthrough therapy designations by the FDA. That's all for today. Thank you for watching Enclave News Network. I'm Gina Columbus.